Students, let us continue with the previous lecture of tools of recombinant DNA technology. In the previous lecture, we have studied about what is biotechnology. It was an introductory lecture. I told you what are the tools required in the biotechnology, what is biotechnology. We have talked about up till enzymes in the previous lecture. So today we will be discussing about the first tool of biotechnology, our DNA technology in detail, that is enzymes. Enzymes, basically, I told you that they also enzymes, they are of two types. If we talk about in biotechnology, so it is lysing enzymes and cleaving enzymes. Lysing enzymes, as I told you earlier also, that lysing enzymes, they are used to break the cell wall, dissolve the cell wall. If it is in case of this fungus, so it will be chitinase enzyme that will be dissolving the chitin cell wall. If it is in case of plant cells, so it will be cellulase enzyme that will be dissolving the cellulose cell wall. So next category of enzymes is cleaving enzymes. Cleaving enzymes, they are of three types: exonuclease, endonuclease and restriction endonuclease. If we talk about exonuclease enzyme, so exonuclease enzyme is that enzyme which removes the nucleotides from the terminal ends, that is 5' end or 3' end. If we have this strand of DNA, so it is going to remove the nucleotides either from this 5' end or from this 3' end, or this 3' end or 5' end. So next is endonuclease enzyme. It is going to cut the nucleotides at the specific position within the DNA strand. Likewise, it is going to cut from any of the part, we can say it can cut the DNA strand from any of the part within the DNA. If we talk about restriction endonuclease enzyme, it is an important enzyme in case of biotechnology, and it is used to cut the DNA strand at a particular sequence. It will recognize the particular sequence, and that particular sequence is the palindromic sequence. It will recognize that palindromic sequence, and it will cut the DNA strand from that palindromic sequence. And it will produce the sticky ends. Sticky ends will be those ends which will be produced after the cutting of after the cutting is made by restriction endonuclease enzyme at the palindromic sequence. Now let us talk about what is the palindromic sequence. If you talk about palindromic sequence, it is that sequence which, if you read from 5' to 3' or from 3' to 5' direction, so it is same in both the directions. For example, AATT. If you start from reading it from 5' end, it is AATT. If you take the complementary strand of it, complementary of A is T, T, A, A. Again from 3' end, T, T, A, A. So if we are going to read it from either of this direction, either from 5' to 3' or from 3 to 5, it is same. So that is your this palindromic sequence. For example, another example is, in a common language we say Nitin. If you write it 5' to 3' or from 3 to 5, so you will be writing it in the same manner. So that is the palindromic sequence which are found in the DNA template. So they are recognized by this restriction endonuclease enzyme. It makes that cut at the particular palindromic sequence, produces the sticky ends, and as I told you earlier also, later on those sticky ends are joined together by the DNA ligase enzyme. Now let us talk about restriction endonuclease enzyme. Restriction endonuclease enzyme, let us talk about what is actually restriction endonuclease enzyme. It is actually restriction endonuclease enzyme is actually the molecular scissor. It is actually molecular scissor which we are using in biotechnology to cut the DNA sequences at particular positions. This molecular scissor, this restriction endonuclease enzyme was first isolated by W. Arbor in 1962, and he even got Nobel Prize for this. So palindromic sequence And the restriction, this restriction endonuclease enzyme is basically of three types: type 1, type 2, type 3. If we talk about the types of restriction endonuclease enzyme, so it is of three types: type 1, type 2 and type 3. If we talk about type 1 and type 3, so type 1 and type 3, they are not utilized in our DNA technology because they need ATP energy for ATP molecule for their functioning, so they are not utilized in our DNA technology. But type 2 is basically used in our DNA technology. That is because for the two reasons. The very first is that it doesn't require ATP. No ATP is required for its functioning. For the cleavage mechanism, it doesn't require any ATP molecule. And the second thing is, it makes cut at both the strands of DNA. Both strands of DNA. So that is why type 2 is preferably used as a restriction endonuclease enzyme in case of our DNA technology over type 1 and type 3. If we talk about nomenclature of restriction enzymes, so let us take an example. For example, we have one enzyme, restriction enzyme EcoR1. EcoR1 is the enzyme that has been isolated from the bacteria that was first isolated. So it has been named, E is the name of the bacteria from which it has been isolated. E is the name of bacterium. So in this case we have Escherichia coli, the bacteria which is used. CO is the coli is the species. R is the strain of the bacteria that has been used, and 1 is the number of isolation, at what number it has been isolated. So that is how the naming of restriction enzymes has been done. It includes name of this species, name of this organism from which it has been taken, name of the species, then the strain, and then the number of isolation. What is the sequence? If we talk about the working of restriction endonuclease enzyme, as I already told you, that the working will be, it will be making the cut in the foreign DNA. The plasmid DNA will be there. Then they both will be joined together with the help of DNA ligase. Now, the DNA ligase will only be working in the case when it will be producing the sticky ends. So means restriction endonuclease enzyme will be working in the case of the production of sticky ends only. Means it will, after cutting, it will be producing the sticky ends. Sticky ends means they will be having the capability of joining together after the DNA ligase will work on them. So that is how restriction enzyme will be working. And palindromic nucleotide sequence, as I already told you, that it is that sequence which is, when you read it